Hello again, everybody. Welcome back. Today we're going to be talking about mitosis and meiosis. These are two processes that are actually quite similar in that they're both processes for dividing cells, but as you're going to see here, they provide us with very different results. Both of these, though, are part of the overall cell cycle, which is the sequence of events that occur in the lifespan of a cell. So the two main events, I guess you could say, of the cell cycle that we're going to be talking about today are mitosis, which is going to be the division of cells to produce genetically identical copies, and meiosis, which is a process of making genetically unique gamete cells that are then used for reproduction. Before we get started on how eukaryotic cells do that, we're first going to talk about prokaryotes a little bit. Bacterial cells divide by a process we call binary fission. And this, compared to mitosis, is a relatively simple process. So in this image right here, the little blue area here, this is our DNA of that bacterial cell. And so the first part of binary fission is that DNA is going to start copying itself. And essentially what's going to happen is a little spot is going to form here that we call the origin of replication. And that's oftentimes just abbreviated as ORI. From there, after that replication begins, you'll start to see those two cells kind of pulling apart from each other. At the end here, you'll see we've got our two distinct sets of DNA, and then ultimately those cells split, and what we end up with is two identical copies of a bacterial cell. In eukaryotic cells, it's a little bit more complex. The cell cycle, remember, is divided into phases. The G1 phase is our normal growth phase. This is our normal day-to-day activities. The S phase is the synthesis phase, where we are replicating the DNA. The G2 phase is just preparing to divide. So our cells are getting ready to go through the process of mitosis, which is the M phase. But again, it's mitosis and cytokinesis. The two of those together make up cell division. So all of these first three phases right here, these are what we collectively call interphase. And then the M phase is, of course, cell division, which consists of mitosis and cytokinesis. The final phase of the cell cycle is what we call the G0 phase, and this is the quote-unquote resting phase. And it doesn't mean that the cell is resting, not doing anything. It just means it's resting from cell division, so it's not actively working towards dividing. But today we're going to focus mainly on the M phase. So also in the cell cycle, it's important to remember there are certain checkpoints that occur. So here we have 4 separate checkpoints, sometimes also referred to as restriction points. So what these serve as is kind of like the brakes on the cell cycle. So these are signals that are basically saying we're going to stay in this phase until something pushes us past that restriction point. And same thing, once we enter the S phase, we're going to be held up by this restriction point until something pushes us past. And every phase is going to have a certain restriction point that you have to get past in order to make it into that next phase. One of the most important phases in the cell cycle is the DNA replication. So there's a couple things that are important to be familiar with here. So this picture is showing us down here at the bottom, if you look at this shape right here, this is a chromosome. And chromosomes, we all know, are made up of DNA, but there's a little bit more to it than that. So if you look at this diagram, here we've got our strand of DNA, and then right here we've got these clusters of proteins, and those are called histones. And when DNA wraps itself around those histones, it makes something called a nucleosome. And so these structures become really important when we start talking about how DNA is going to be transcribed and translated later on. Once that DNA has wrapped itself around those histones to form nucleosomes, then those nucleosomes start to coil up onto each other, and this makes something that we call chromatin. So DNA wrapped around these histones that are all coiled up together, those make up chromatin, and that chromatin then condenses to make our chromosome. And what holds our chromosome together is a little protein right here in the middle called a centromere, and that's going to hold the whole thing together. So I'm going to go through this really quickly. The individual phases of mitosis are not important for you to memorize. It's more the overall process. So I'm going to speed through here really quickly. Again, this is stuff that you should have seen at least a couple times in the last 4 or 5 years in your science classes. So to start off, over here on the left, this is a cell that's in interphase. You can see in these images here, DNA is stained red while the cytoskeleton is stained green. So this is just our normal cell in interphase. As prophase begins, you'll notice that the DNA is starting to become a little bit more condensed, and those chromosomes are starting to become visible. And you can also see here our nuclear envelope is starting to break apart. So the actual nucleus is starting to break down a little bit. The next phase is called prometaphase, and so now here you can see those chromosomes are becoming more visible, and then you can also see these green cytoskeleton filaments starting to form what we call the spindle, which are these little fibers that attach themselves to the chromosomes. In metaphase, now we've got all of our chromosomes lined up down the middle of the cell, and again you can see these green lines coming off here, and in our stained image up above, those are these spindle fibers forming that are going to help these chromosomes separate. After metaphase is anaphase, and now you'll see these spindle fibers are shortening and our chromosomes are getting pulled apart from each other. One little piece of vocabulary here that we haven't covered yet. So if this X represents a chromosome, each half of that chromosome is an identical copy of the other half. And so each side of a chromosome is called a chromatid. And so during anaphase, we have separation of sister chromatids. So we're actually splitting this chromosome right down the middle, and then what you'll end up with is this half going this way, and then this half going that way. So we're just splitting that one chromosome in half. And then the last phase of mitosis is telophase, and this is where now you're starting to see the nucleus forming over here and a new nucleus forming over here. Notice we're still one big cell. A lot of people have a misconception about this, but at this point, mitosis is over. Cell division has not finished, though. Cell division occurs when we have mitosis, followed by something called cytokinesis. Cyto refers to cell, kinesis refers to movement. So cytokinesis literally means cell movement. And what you'll see here is these two cell membranes will kind of pinch off in a process that we call cleavage. And that will separate then into two new separate cells. So cell division has a lot of different uses in our body. Mitosis is mainly used for repairing and replacing cells. But cell division can also be used for reproduction. For organisms that reproduce asexually, they can basically just divide and create clones of themselves. So bacteria can clone themselves. There's a lot of species of plants that are able to just reproduce asexually. And while this is good, it allows organisms to reproduce. The downside to it is there's no genetic diversity being generated. You're just producing copies of each other, producing clones. Sexual reproduction, on the other hand, that involves specialized cells called gametes. And gametes are the sperm cells and the egg cells. And these are very specialized cells that serve no other function other than for reproductive purposes. And the good thing about sexual reproduction is it increases genetic diversity. So we're taking all the different combinations of traits that are out there and we're making entirely new combinations. And when we start talking about evolution, we'll really get to the nitty-gritty of why that is important. Okay, so this diagram here represents the normal lifespan for humans and other vertebrates like us. And so this shows a diplontic life cycle, which means most of our life cycle is spent in this diploid zone. So diploid cells are also referred to as somatic cells. These are all the cells that make up our body. And we have 2 copies of every chromosome. So in humans, our diploid number is 46, which means we have 23 pairs of chromosomes. In those pairs, so let's say here's one chromosome. Whoa, that's a really bad chromosome. And then here's another one. So let's say both of these represent chromosome number 1. The red one over here would be the one that we got from our mom, and the green one over here would be the one that we got from dad. And together, these are what we call homologous chromosomes, or they are homologs to each other. And what that means is they have corresponding genes found on them, but not necessarily the identical genetic information. So for example, the chromosome I got from my mom, I might have the gene right here for blue eyes. I'm just going to make this really simple. From my dad on this chromosome, the gene for eye color is in the same place, but maybe I got the gene for brown eyes from my dad. So they're homologous in that they have the same genes on them, but they have different versions of those genes. And we'll get to that a little bit more when we get further into genetics here. But going back to the diagram over here, most of the cells in our body are diploid. And then through the process of meiosis, we produce haploid cells. So these are cells that only have 23 chromosomes. They just have 1 copy of each pair. And so in males, the cells that are going to be produced are sperm, and in females, they're going to be eggs. When the sperm fertilizes the egg, they come together to make something called a zygote, which is the single cell that we all start from. And those zygotes are diploid, as well as the rest of the cells in our body, and they just keep going through this cycle over and over. Okay, so what happens during meiosis? This is what we call a reductive division. And what that means is we're going to start with cells that are diploid, and by the end of meiosis, we're going to produce haploid cells. So we're going to reduce the amount of DNA in each cell. Meiosis 1, there's 2 overall divisions that happen in meiosis. There's meiosis 1 and meiosis 2. Meiosis 1, this is where really the most important stuff happens during the process. So in meiosis 1, our homologous pairs are going to line up side by side, which they don't do in mitosis. In mitosis, you'll see these chromosomes kind of lining up in the cell in the middle like this. Actually, let me draw it this way. So here's chromosome 1, here's chromosome 2, here's chromosome 1, here's chromosome 2. So again, these 2 are from my mom, these 2 are from my dad. In meiosis, instead of lining up like that, they're going to line up like this. They're going to line up side by side, and they're actually going to be touching each other, which doesn't happen in meiosis or mitosis. And so in meiosis 1, these homologous chromosomes are going to line up, and then we're going to have separation of those homologous pairs. But what's going to happen is the individual chromosomes are going to remain intact. And then in meiosis 2, we're going to have the separation of sister chromatids. The big key here that's different between meiosis and mitosis is that in between meiosis 1 and meiosis 2, there's not any DNA replication. That's where we get this reductive division. So our cell is going to go through the normal process. The DNA is going to copy itself. We're going to go through the first division. And then after that first division, we're going to go straight into our second division without copying the DNA a second time. So in this process of meiosis 1, when those chromosomes line up side by side, this is where we get our increase in genetic diversity. And where that comes from is this process called synapsis. So when these chromosomes line up side by side like this, that's called synapsis. There's another term that you'll hear called crossing over, which is basically the same thing. But what happens during synapsis is these two homologous chromosomes line up, and then anywhere here that you see them crossing over with each other, those are what we call chiasmata. The singular is chiasma, plural is chiasmata. So everywhere these chromosomes form these chiasmata, there's going to be an exchange of genetic material. And so what you'll actually see is, look at this next picture, here's our two homologous chromosomes. They line up with each other, they cross over, and there's an exchange of genetic material. So now what was once on this red chromosome is now moved over onto this blue one and taken its spot right there. And then I don't have blue, so I'll just draw with green or with white, and that one crosses over and exchanges right there. When they're crossed over like this, this structure is called a tetrad. And tetra, that prefix means 4. This is just referring to the fact that there's 1, 2, 3, 4 chromatids that make up this tetrad. So again, I'm going to run through this really quickly like I did with mitosis. What's most important here is our big picture. So early on in prophase 1, our chromosomes are just becoming visible. And by the end of prophase 1, moving into prometaphase, now we're seeing those chromosomes lining up next to each other and forming those chiasmata to allow the crossing over process to occur. In metaphase 1, now you'll notice these chromosomes are they've lined up in the middle of the cell, and by anaphase, they're pulling apart. But what I really want you to see here, let's look closely at this guy right here. Notice that now these chromosomes, it's not entirely DNA from mom or entirely DNA from dad. Each homolog in these pairs is now a shuffled up mixture of mom DNA and dad DNA. This is the only time in our body where we have chromosomes that contain DNA from both of our parents. And then by the end of telophase 1, now the DNA is starting to loosen up again and uncoil, and ultimately here we're going to end up with 2 cells. And then meiosis 2 is almost exactly the same as mitosis. So our nuclear wall starts to break down, the chromosomes line up in the middle of the cell, and then the sister chromatids are pulled apart from each other. And by the end, now we have 4 cells that are haploid, that are genetically unique from each other. So if we really summed it all up, if we said mitosis versus meiosis, mitosis takes place with 1 division, whereas meiosis takes place in 2 divisions. In mitosis, you start with a diploid cell and you end with 2 diploid cells. We'll say 1 diploid cell becomes 2. Here we have 1 diploid cell becomes 4 haploid cells. That's 1N. Mitosis produces genetically identical daughter cells, whereas meiosis produces genetically unique daughter cells. And finally, mitosis is used in the division of somatic cells, whereas meiosis is only used for producing gametes. So I know this is a lot of information. We went through this really quickly, but over the next few lessons, we'll reinforce the importance of all of these different things. And in the next one, we'll actually talk about what happens if there are mistakes during this process. But for now, that's mitosis and meiosis, and I hope you found that helpful.